So imagine that you just woke up from a nap and now you can't breathe through your nose. You think you have a common cold, so you take a bunch of drugstore medicine, Tylenol, Dayquil, NyQuil, Robitussin, more drugs. It's been a week, nothing works. The next thing you know, your mom is dragging you to the doctors. There they make you take a bunch of tests, examine your lungs, throat, and tongue, and the doctor says, you have something, bronchitis, something. You're going to need a prescription of antibiotics for the next 5 days. And also take in an inhaler with medicine that will clear up the congestion from your lungs. Have you ever wondered what is in those drugs that makes our symptoms go away? What exactly do they have in them that works on our body in certain ways and improves our situation by altering chemical or ratios in our biological system? It's the chemical composition of those drugs that reacts with our body in certain ways and creates a desired outcome. Chemistry plays an essential role in the field of medicine. Most of the drugs used for treatment, cure, prevention of a disease, or disorder are made of some chemical. The study of these chemicals in medicine is quite fascinating, and I'm going to take you on a tour of how chemistry and medicine go hand in hand and how it affects our bodies and drugs we use to cure health problems. Our bodies are wholly made of chemical substances, and all our bodily functions depend on chemical reactions. Chemical reactions allow us to digest our food, keep us warm, and supply us with muscular energy. The probability is very high that even the impressions of our senses and the thoughts of our brains, as well as the way of conveying these thoughts through our nerves, all have something to do with chemical reactions. So basically, you watching this video and processing what I'm saying is a chemical reaction. In short, the human body is an intricate chemical machine, and its well-being, the illness, are basically connected with the coordination of a variety of complex chemical changes. The first thing we're going to discuss is the 3 different ways chemistry can be used in medicine. One of these is concerned with the components of things. This kind of chemistry is called analytical chemistry. Another type of chemistry involves not only what to find out what things are made of, but to also find out how they are put together. This type of chemistry is known as structural chemistry. Lastly, another helpful chemistry is more recently developed and known as physical chemistry. It's basically on the borderline of physics and chemistry, but it is more concerned with the relation of energy to material. The physical chemist must know not only what things are made of and how they go together, but also what energy is used putting together and what energy is released when a substance decomposes. All of these types of chemistries all go together and are kind of like the building blocks of one another. To elaborate more on analytical chemistry, the human body is an intricate machine of chemicals. The analytical chemist is able to tell us the composition of each one of the substances that compose the human body. He can not only say what elements are present, but also the exact amounts of them with a lot of precision. He can analyze food and bodily fluids, which can determine the relation between the two. This is all obviously of great value in cases of disease because it gives us excellent clues to the manner in which the various functions of the body depart from the normal, and so gives important aid in diagnosis and the suggestion of suitable treatment. To break it down, a medical physician needs to know the actual composition of the drug, understand the chemical nature of the drug. What does that mean? Different medicines have different chemical properties. like acidity, alkalinity, it's either oil soluble or water soluble, and it can also be polar or etc. For example, an acidic drug will be better absorbed in the stomach, while alkaline drugs absorb well in the intestines. Analytical chemistry made it possible for us to know which drug is better suitable for us in certain organ. Lastly, a physician needs to be able to predict how a specific drug will react with a specific part of the body and another drug the patient might be taking, because not all drugs are compatible with each other. If the patient has a gastric ulcer, the doctor might prescribe an antacid like calcium hydroxide, and also an antibiotic like tetracycline. The only problem is that these two drugs aren't compatible with each other at all. Tetracycline will form a complex with calcium. Why is that? Antacids reduce stomach acid to relieve heartburn, sour stomach, acid indigestion, and upset stomach. Antibiotics are medicines that help stop infections caused by bacteria. Antibiotics are usually taken orally as well. In order for oral antibiotics to be effective, they must be absorbed from the gastrointestinal tract, basically the stomach and spleen, make their way into the bloodstream and be delivered to the infected area. This antacid has calcium and in general milk. A substance containing calcium is not supposed to be taken with antibiotics because the calcium in milk binds the antibiotic and prevents gut absorption. Moving on to structural chemistry. Structural chemistry started with the discovery that two substances might have the same chemical makeup or percentage amount of exactly the same elements. However, they could both be completely different things based on how they're arranged. We can get a better idea of this concept through the atomic hypothesis. For example, if the smallest particle of any given compound substances are built up of still smaller atoms of the various elements concerned, it is clear that we can have many different arrangements of these atoms. And it is reasonable to suppose that the particular arrangements might make considerable differences in the nature of the resulting compound. Structural chemists depend on two ways of working. They either analyze the way a compound splits up and finding out what group it decomposes into, or puts the substances back together with the chemical debris it left behind, kind of like a puzzle, exactly like an experiment. It is obvious that structural chemistry is vital to medicine because you need to know what elements make up every part of the body to be able to treat diseases and sicknesses. The drugs taken to cure these illnesses have to be able to react with the body in a way where it won't hurt the patient. Moreover, in the case of our food, the arrangement alone of the atoms may make all the difference between nourishment and poison. The knowledge of the atomic arrangement of the various substances of the body also helps further the study of physiology, pathology, and hygiene. With this, in the future, scientists might be able to reconstruct worn-out organs entirely out of pure substances in the lab, as well as destruct malignant germs, tumors, without doing any harm to the body. We might also be able to get artificial immunity from diseases like smallpox. The possibilities are literally endless. Now, there is no doubt that energy is the immediate cause of every action in the known universe. All organic life depends on chemical energy. That's why the study of chemical energy is very important. Physical chemistry has to do with the relation of each of the various kinds of energy to chemical change. It deals with the acting driving forces which make life possible. And in each of its many aspects, it brings new intelligence to show us how the human body works. Physical chemistry treats, among other topics, the chemical relations of the changes from solid to liquid and from liquid to gas. It discusses the nature of solutions and mixtures of all kinds. As a living body is composed of solids and liquids and depends upon the gases of the atmosphere for promotion of the chemical changes, keeping it alive, and as solutions and mixtures are present in every cell, the laws and theories of physical chemistry are intertwined with every fact of physiology. Again, physical chemistry deals with the relation of heat to chemical change. Moreover, physical chemistry studies the effect of changing temperature upon the speed and tendency of chemical action. This is very important in the study of fevers, other abnormal conditions, like hypothermia, as well as how our body stays at a constant temperature. Within all this lies the connection between chemistry and electricity. Our branch of this subject is photochemistry, the chemistry of light. Research in photochemistry helps a lot with understanding the changes between leaves of plants from being in the sunlight. With only the help of light, nature builds food for every animal, including us. Until we understand the growth of vegetables, we can't even hope to understand the growth of us animals. These three types of chemistry are the very basis of how we treat diseases, in the study of nutrition and much more. Other ways chemistry is used in medicine is for sterilization and sanitization. Most chemicals like alcohol, phenol, acids, and aldehydes are used for disinfection. Knowledge of chemistry helps to know how the substance possibly kills the microbes. Microbes are bacteria that cause disease and infection. Depending on that, the physician can decide which type of sterilization can be used for different purposes. For example, if you watch Grey's Anatomy before, you know that right before they cut someone open, they pour betadine all over the area. Betadine is an antiseptic used for skin disinfection before and after surgery. It disinfects the skin of the patient as well as the hands of the surgeon, especially in traumas, because there isn't always enough time to scrub. Many diseases or disorders are easily diagnosed by simple biochemical tests. Biochemical tests are the tests used for identification of bacteria species based on the differences in the biochemical activities of different bacteria. These differences in carbohydrate metabolism, protein metabolism, fat metabolism, production of certain enzymes, ability to utilize a particular compound, etc., help them to be identified by the biochemical test. Some identification based on the structural tests are not possible, because even if different bacteria have different structures, they still can't be seen from under a microscope. Therefore, ultimately, the identification of bacteria is mostly based on the differences in the biochemical activities. Some biochemical tests include the indole test, the methyl-red test, Vogt-Proskauer test, cyclic utilization test, triple sugar iron test, carbohydrate fermentation test, and many more. Each species of bacteria has a well-defined set of metabolic activities, different from all other species. These biochemical fingerprints are properties controlled by the bacterial enzymes. The bacterial enzymes can be grouped into two different categories: the intracellular enzymes, also known as endoenzymes, and the extracellular enzymes, or exoenzymes. Intracellular enzymes function inside the bacteria cells. Their main job is to synthesize new protoplasmic substances and production of cellular energy from simple substances spreading into the cell through the cell membrane. The enzyme is necessary for cellular function and is the basis of cellular metabolism. Metabolic products are formed and taken out by the cells into the environment. These end products help with the identification of bacteria. Extracellular enzymes are the enzymes produced by the bacteria cells to the surrounding to hydrolyze, which means to break down by chemical reaction with water, high molecular weight or complex substances, such as proteins and fats, which cannot spread the bacterial cell membrane. The enzymes hydrolyze them into their respective categories. Proteins are broken down to amino acids, polysaccharides to glucose, and triglycerides to fatty acids. The low molecular weight simple substances so produced can spread through the cell membrane into the bacteria cells, where they are used with the help of intracellular enzymes to identify the bacteria. I know that's a lot of information, so an easier way to remember those two is by the prefixes. Intra in the word intracellular enzymes refers to inside the cell, while extra in the word extracellular refers to outside the cell. One test I found particularly interesting is the glucose broth with Durham tube test. The purpose of this test is to see an organism's ability to ferment the sugar glucose, as well as its ability to convert the end product of glycolysis. The breakdown of glucose by enzymes releasing energy in pyruvic acid. Basically, the test shows how well your body manages glucose after a meal. Glucose is a type of sugar produced when the body breaks down carbohydrates consumed in food. Some of the glucose will be used for energy, while the rest will be stored for future use. This was a test commonly used when trying to identify gram-negative enteric bacteria. Bacteria that live in our intestinal tract, which are all glucose fermenters, but some of them also produce gas. The solution used for the test also contains the pH indicator, phenyl red. If an organism is capable of fermenting the sugar glucose, then acidic byproducts are formed, and the pH indicator turns yellow. The type of bacteria that can ferment glucose are Escherichia coli, better known as E. coli, Proteus mirabilis, and Shigella dysentery. Pseudomonas aeruginosa is a non-fermenter. The end product of glycolysis is pyruvate. Organisms that are capable of converting pyruvate to formic acid and formic acid to molecular hydrogen and carbon dioxide via the action of the enzyme formic hydrogen lyase emit gas. This gas is trapped in the Durham tube and appears as a bubble at the top of the tube. E. coli and Proteus mirabilis are both gas producers. Shigella dysentery ferments glucose but does not produce gas. Whoa, that was a lot. Let's go back and see all of the things we learned today. We learned about 3 different types of chemistry that are used in medicine: analytical chemistry, structural chemistry, and physical chemistry. Next, we talked about sterilization and how a big part of it plays in the art of surgery. Lastly, we went in depth about biochemical tests and different things we use and look for through those tests. Obviously, there's a lot more to be learned from this field and so many amazing things left to be seen. I hope you enjoyed the video and it was helpful to you in a way. Thank you for watching.